Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Jeremy Bar. He's the head of the bacteriophage biology research group. He's also a lecturer at Monash University in Australia. And the purpose of the call today is he's also going to be a contributor to the virus book that I'm going to be putting out in the next couple of months here. So again, Jeremy, he's going to be a good contributor. He focuses on bacteriophage, viruses that infect bacteria. But I'm also going to ask him questions about, viruses that infect people, etc. So, Jeremy, thanks for coming.
Yeah, thanks for having me.
Yeah. Well, just to start with, before we get into the questions about you, what got you into studying viruses? What attracted you to it?
Yeah, I think I was really lucky in that I knew from an early age what I wanted to do. I think as a kid, I was always interested in science and actually fell in love with insects when I was sort of maybe 8 to 10, and I was obsessed with them and reading all sorts of books. And then as I got older and moved into university, I drifted towards microbiology and eventually settled on bacteriophages. And I think, looking back, I would arguably say that bacteriophages are similar to the insects of the microbial world, and that they're incredibly diverse, they're unique, they're extremely exciting, and they've always got sort of different surprises. So I've always been really, really interested in the sort of weird or quirky aspects of biology. So I think that's what drew me to viruses.
And what's your research about currently?
Yeah, so my lab at Monash University, we look at bacteriophages. So as you mentioned, bacteriophages are viruses of microbes, specifically of bacteria. And our lab looks at phages in this concept of tri-partite symbioses, which if I was to break it down, I would say that within the human body, you have your microbiome, specifically your bacterial aspect, and that these have their own viruses, being bacteriophages. But those phages don't just predate on their bacteria; they also interact with the human host as well. So this concept that virus, bacteria, and metazoa, or human host, have all of these intricate symbioses is sort of a focus of the lab. So looking at phages in the context of bacteria, but also within the context of the human and mammalian.
So there are bacteria that they may not pathogenically interact, but they interact both with bacteria and with eukaryotic cells at the same time?
Absolutely. Yeah, for sure. I think it's a
really,
it's a really, I think when you think about it really logically, it makes complete sense, right? Your body has so many mechanisms and ways to sort of sense and interact with the environment. And these phages are such a major component of the human body that I think it's, would be amiss to think that your body doesn't sense them or doesn't respond to them or interact with them in lots of diverse ways. So our lab is sort of uncovering lots of different mechanistic ways that phages can directly interact with the mammalian host.
I think probably most people are afraid and they don't want them to interact with us because they think it'll be bad.
Yeah, I can definitely understand that viewpoint. But I guess, a lot of our research is trying to shy, we do some pathogenic research, but I guess that will be a major point that I'll sort of make in this interview, is that these pathogenic interactions are such a small component of virology that, the vast majority of interactions are actually beneficial. So there's lots and lots of good things and beneficial ways in how these viruses interact with our own bodies that, if we can begin to understand, then we can begin to apply them.
So jumping ahead a little bit, just because you brought this up, if let's say cells in my gut are infected by a virus,
yep.
How do you think that's going to result in a change of the local microbiome near those cells in my gut?
Yeah, so many different ways. And I guess the first key point is whether you're talking about a eukaryotic virus, so a virus that can infect and replicate within the gut cells, or if you're talking about another virus, say a phage. And I guess I'll I'll keep coming back in this interview to the phage perspective, but I can speak more broadly to the eukaryotic viruses. So to give you an example, so a viral infection of your gut cells can change all of the metabolism and the processes that are happening within that cell, and that can directly influence the receptors and the molecules that the cells will display on their surface, and even their secretions. And so one of the big research areas that our lab is looking at is the mucus layer in the gut. And this mucus layer is critically important for your gut microbiome. It provides a food source, it provides a structural resource for them to grow on. And so there's been evidence that viral infections, not only pathogenic, but some symbiotic or off-target infections, or interactions is a better word, can actually change that mucus structure, and that can actually change the species and the growth of specific gut bacterial species. And this can have knock-on effects to the function of your gut, be that metabolism, inflammation, and many, many other complex ways.
How would you guess that the action of a phage versus a eukaryotic virus, let's take the example of in the gut,
Yep.
How would you think that both would affect, again, the microbial constituents that are local to the cells that are being affected?
Yeah. So let's break it down. So I'll start with the phage angle. So obviously the phage can directly affect the bacterial species. They can have top-down predatory, but looking at that direct interaction, and a lot of our research investigating this symbiosis of phages. So there's an example that I'll touch on later, where we show that phages bind layer, and binding there the increased interactions with bacteria. And another aspect of our work is looking at engulfment and so this actually happens all the time. Back of the 30 billion phage uptake by cells average human. And so your gut cells are pulling those phages. Those phages persist, they traffic, and I think there's lots of molecular ways eukaryote and be that genetic information interaction, and they can change metabolism that can then feed back to the gut microbiome. The positive feedback loop. Phages are transmitting the gut cell, which then feed back a signal back to that microbiome.
That's amazing. Wow.
Yeah. So that's sort of a more of a sort of system level view that my lab's trying to break down in different specific parts.
Well, I'm going in a different order for the questions here, because, I'm focusing on your expertise, I think, first. So if phages mediate behavior of, obviously bacteria in our cells, in the virion stage, I could see, okay, they probably don't really mediate any behavior, but once they're inside a cell, it seems like they're like sitting at a control panel now, and they're able to use the cell to accomplish what they need. So do you think that, once viruses are inside cells, are they able to use the cell's machinery to do quorum sensing? Like if I'm imagining a virus sitting in the control room of a given cell, can it communicate with other cells and say, hey, are you infected by a virus like me? And again, can they coordinate behavior by using the cell's machinery? Yeah,
they definitely can. That's a really the control. So yes, they absolutely can. And again, on phage aspect. So this has been a somewhat some recent discoveries in the quorum sensing, and how bacteriophages will co-opt bacterial systems to actually regulate when they lyse and when they kill a host, and they persist as a lysogen infection. And so specifically phages will, there was a molecule named arbituum, I think it was, which is a quorum sensing produced by a bacterial cell, which a phage has co-opted, and they use it to sense how densely populated the bacterial community or ecosystem population is, and they use it specifically to time when they're going to enter the lytic cycle and produce more virions, or when they're going to stay as a lysogenic. And it's sort of sensing that extracellular bacterial population to maximize their reproductive success. And if they sense very few bacteria, well, it might be better to stay as a lysogen, ride out your conditions all through. Or on the flip side, when those increase, you can actually sense bacterial high density, and thereby opportune time to enter into.
That's awesome. Do you think that they're able to sense not only the relative number and concentration of bacteria, but do you think they're able to also sense if they're infected as well with the virus, just like them?
Yep, they definitely can. And there's some incredible, both molecular ecology mechanisms that these phages. And maybe one example I'll give you is the super infection exclusion mechanism. So this is when a bacterial host is already infected by, is typically in a lysogenic infection. So a phage is sort of persisting in its bacterial host, riding it out, hope for better days before it enters the lytic cycle. And these super infection exclusion mechanisms will actually block other phages from infecting that bacteria. So From the phage's perspective, it doesn't want another virus coming in stealing its host for its own use. And so it will produce a range of proteins or enzymes that will either block the injection or penetration of new phage genome cell or actively degrade those. And it also can delay those lysis events. So from a lytic phage that's actually replicating, there's a system whereby every subsequent phage infection that phage won't take over the cell due to these exclusion mechanisms, but will be sensed by that infecting phage and will actually delay lysis. And so you can delay lysis for hours, days, and continue this going. And from the ecological perspective, the phage is infecting and it's filling that cell with new virions, but it doesn't want to lyse if there's a huge number of viruses already out in the environment, because it would just be competing against them. So by continually sensing how many infections are sort of coming into this host cell, it can choose to delay, and wait until that cell's no longer being infected, and then lyse to optimize its reproductive chances.
Yeah, this goes to another question. Why is there a latency period? And latency could be initial infection to lytic behavior, initial infection to lysogenic or lysogenic to lytic, and back and forth. And it seems like you're saying that that's governed by a continual checking and reading of the environment and seeing when the conditions are right.
Yep. Absolutely. So, the latency period, I find it really fascinating because it's almost like a metamorphosis, right? You could look at, going back to that insect where a caterpillar changes into a butterfly, and it's a similar change, right? You go from a structural virion persisting in the environment into this quasi-molecular state where the virus exists purely as genetic information that's sort of controlling and manipulating the cell. So it's a really huge and rapid metamorphosis of that lifestyle. And as to why and when and how they exist in that in period, there's a huge range of reason and mechanism, and I think it all comes back to optimizing that virus's replicative success, not only in the bacterial host that it's in, but also in the ecology in the larger relation. Incredible ways that the phage can drive and manipulate that cell, whether that's producing virions or whether that's giving the cell benefits to then increase the virus's chance of persisting.
Yeah, I saw a paper where I had to find the reference, but this particular virus, they noticed that created tons of virions, but a lot of them were empty. And I wonder if that was a decoy system, whereby I think it was a eukaryotic virus. Then the host would see a lot of these virions, maybe expend its immune response energy on trying to, engulf and get rid of them. And then there would be a few select ones that do have the genetic payload that would be used to carry out what's needed. Any thoughts on if why that would happen?
I could definitely see a benefit to it. And I might take the other example in that the opposite point of view, in that if you're looking from a bacterial perspective, evading these viral infections is hugely important, and you can make the same analogies to our own cells and against eukaryotic viruses. But some of these bacteria will produce, small vesicles as almost decoys to absorb viruses and decrease the chance that the bacterial cell will be infected. I can't think of a specific example where a phage produces unpackaged virions. I know there's examples where you'll have sort of predatory viruses which will try to parasitically package the virion of another infecting phage with its own genome. And there's other mechanisms where the phage can package bacterial DNA or other molecular information. But yeah, that's an interesting viewpoint. I don't know if I've seen that reference either.
Wait, so are you saying that phage can, inside a cell, I would think the only thing they would do is package up new virions that are faithful to what they are.
Yep.
But are you saying there are instances where a phage seems to direct activity where it does the capsid and then it stuffs it with different material and sends it out?
Yep, absolutely.
Really?
Yeah. So this is one of the biggest mechanisms of horizontal gene transfer induction, where some phages just naturally do this on accident, where when they're infecting a cell, they won't completely degrade the bacterial host DNA. And on occasions, they'll package bacterial DNA in some virions. And now you have a virion, but it doesn't contain any phage information, it contains bacteria. So if that virion infects a bacterium, it can actually horizontally transfer that genetic information. So that actually happens quite commonly. And there's some recent papers showing that certain bacteria are actually super spreaders and really maximize this mechanism. And there's also examples of parasitic viruses where a virus will sit in a cell in a lysogenic infection, and this virus has no structural gene. There's no mechanism of escaping that cell. Yet when another virus infects that cell and it starts replicating, it will produce structural protein, but this parasitic virus will actually steal its thunder and package its genome into those capsids. So it can't infect on its own, but when another virus infects, it almost takes it over and steals all its work for itself, so then it can go out and spread into new hosts.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
That's crazy.
Yeah.
Each thing you tell me leads to the next question without, before I even ask it. So that's amazing. So I saw a video and, I read a paper on how I think it's a T4 phage enters into E. coli, and it was one of those moonlander looking phages, with the collar and the base plate and all that. And it was really sophisticated in how it entered. But what I wondered, what if you were able to create a denucleated bacteria, or one that was empty except for the outer membrane? Would a phage land and dock and bind and enter, or do you think it would sense something is amiss and stop before it fully entered?
It's a really good question. I think my first answer would be that it would absolutely absorb and infect that I'll call it a decoy cell, right? Because bacteria do produce small vesicles that don't contain any genetic information as a decoy to sort of sequester phages. So they can do that, and they will infect those decoy, and then it's sort of injected. The virus DNA gets in there, and it's like, oh crap, there's no ribosomes, there's nothing here that I can utilize, and it's sort of a dead end. But on the other sense, phages mechanically sense their bacterial host, and so that T4 example is really it's my favorite virus, T4, and so it has these tail fibers which mechanically wrap up and sit around its head. You always see them down in that Spacelander image, but they actually wrap up and sit around their head, and then that's their sort of highest energy state, if you will, and then they'll sort of snap down. And they'll reach out and they'll sense the environment. So they'll actually snap down and tap on a bacterial cell. And it's thought that tapping can actually sense whether or not a bacterial cell is active. I'm not exactly sure on the mechanisms there. I believe they're sort of maybe sensing, the electrostatic potential or differences on that cell. But it's thought that they can sense whether or not a cell is actively replicating, or if it's sort of a dead or persisting remnant of a cell fragment. So there's a bit of both there. They've got some really incredible ways that they operate.
So underlying all this, a lot of the scientists I speak to, they believe in the modern synthesis, neo-Darwinism. It doesn't seem like you do. I personally think things go way beyond that. Without directly answering that, I mean, do you think viruses are alive? Are they contingently alive when they enter a cell? Are they alive the whole time?
Yeah, I think they're alive the whole time. I think this argument of viruses can't be alive, they have all of these hallmark characteristics that we define as being alive. I would even sidestep the environment and say that, the argument, and say that viruses are life, right? They are, if not the most, they're arguably the most successful life form on the planet, right? There's more viruses than anything else. So they are at the, one of the most basal forms of life as we know it. So this whole argument of, are they alive, are they not? It really doesn't matter, right? They're life, and they are everywhere, and they control and influence almost everything on the planet.
Yeah, amazing. So you've given examples of when a virus infects a cell and then it's able to keep out other infective viruses.
Yeah.
Do you see any instances where it's not just, one virus, one cell that infects? Are there ones that require multiple viruses, let's say of the same species, in order to coordinate entry into a cell? Or is each virus alone capable of entering or not entering?
Yeah, that's really good. I would say that, most of classic biology says that it's each virus is only, is responsible for its own infection and its own entry into a cell. But this is the amazing, this is one of the most exciting things about virology, is I think the viruses break every single rule of biology that we make. You can find an example of where they completely break that rule. And so there are examples of viruses that are completely dependent on other viruses to replicate. And I gave that example of what we call a sort of cryptic phage, which lives in a bacterial host cell. And and it can't replicate on its own. It's got to rely on another virus to sort of piggyback. There's another phage example of P2, P4, where one of these phages relies on the other one to actually cause the infection and package and transmit. So you actually have to use both of these viruses together to propagate. So there are definitely examples where they require other viruses to reproduce and continue on with their life cycles.
Yeah, I imagine a phage, attaches to a cell, and then other phages are attaching to the cell, and maybe through the membrane, through some kind of like transconductance or other signaling, they can coordinate action. Maybe, again, two of them would be near each other and again coordinate entry, etc. I wonder how far this goes. I guess we don't know yet.
It's actually another thought on that. So when a virus does infect a cell, phage infects a bacterial cell, if that enters into the lysogenic state, it can actually cause a process known as lysogenic conjugation, where genes encoded by the phage will integrate into the bacterial, and that bacterial host now has access to that genetic information and can change the phenotype of this can happening. And so theoretically, you could change the structure or the surface of that bacterial cell, which would then open and allow for another phage infection that wasn't previously able to infect that. So I could see systems where you can build could allow additional viruses to predate on a bacteria due to the...
See, this is the weird thing. Where is the life in a cell? Where is the life in a bacteria? Where is the life in a virus?
Yeah.
If it enters and it's just genetic material, I mean, where is the life in it? It's strange.
Yeah, no, for sure. And I completely see that argument. But, genetic material arguably is the most basal codes that life does. Everything that happens in our own cells and our bodies is driven by that genetic. And so I really take that point of view that life can exist as that genetic. And that's true, right? You can, we're right now in this synthetic biology revolution where you can print DNA and you can insert that DNA into a functional lipid environment, create life, if you will, as just viruses do, right? At the most basal level, they transport genetic information, albeit or arguably life, across space and time, stable beyond packages.
Do you think viruses have a group identity? Do they know self? Meaning, these other viruses, their sequence is close enough that I consider them, me. Just like, bacteria form biofilms, probably preferentially with their own strain at first. Do you think viruses, again, have a sense of self, and do they have a group identity?
That's a really tough thing. I don't know if I have a I could probably on either side. I would imagine that they don't, that for the most part that they are individualistic and that they require the success of their own information and propagation of their own sort of virions. But there are examples of cooperation, and I think quorum sensing is maybe a good example of that. So yeah, it's really tough. I could almost take either side of the argument. One example that I was asked to do. So I was actually involved in in this graphic novel that we wrote with a group of friends who run this sort of science communication company. And we had to do this. We had to give the viruses anthropomorphize them and give them a way of speech. And so what we came up with was that these viruses would speak it as a hive mind. So they would, and in the book they communicate as this whole, collective swarm of phages that come in and so that's how we gave them a voice in this book, which was a cool sort of thought experiment. But I'm not sure. That's a really good question. Yeah.
Is that book available, or is it just privately between you and friends?
It's called The Invisible War, a tale. It's a graphic. It's set in World War I, and it was the story of a nurse who was treating a patient suffering a Shigella gut bacterial infection. And then it's sort of this double story. One set in World War I in sort of battlefields with us treating these soldiers, and the other one set in the battlefield, the gut, where the Shigella actually get in the gut and they get an infection, and the phages sort of fight that. I'll send you a link to it.
Okay, yeah, that'd be great. I've heard of a definition of a quasi-species. I mean, what does that definition mean to you? And if, do you think that, again, we're seeing examples of viruses essentially competing, at least inside of a cell, or parasitizing each other. But I guess there has to be some identification of self and other, otherwise they wouldn't be this competition and this parasitism. But again, what are quasi-species?
Yeah, quasi. I don't know if I have the definition for it. It's this sort of continually sliding the definition, right? Where do you draw on the line of this is its own individual species and where is another, right? You could if you go into the genetic information, we can put percentages on it, right? We can say this virus is 98% similar to this other virus at a DNA level. Does that define a species? I mean, we've put arbitrary measures on that, right? I think in some bacterial sense, anything above or below 95% is considered a different species. Does that hold true for a phage? Is that just an arbitrary setting? It probably is, but it's something that we need to use to start to understand the microbial diversity. But it's a really cool argument. At what level does a virus begin to compete versus co-opt that genetic information based on its similarity? And I don't know if there is an answer in that, right? You could look at it from a protein level and see, well, mechanistically, are these proteins similar enough that they do the same function and that they can be synergistic, or when do they start competing? And then to throw a spanner into the works there, you've got to consider recombination. And I think viruses exist right at this melting point and mixing point of recombination, where they can very quickly co-opt other viral genetic information and create entirely new species. Yeah,
what's an example of an amazing co-option by either a bacteria of a viral ability, or a virus of a bacterial ability, or eukaryotic cell of a, et cetera?
Yeah. So, I think I saw this question, had a cool thing up. So this work was done by an old colleague of Nick Shakun, SDU now. What he found was, what he was looking at is these tube worms. So these tube worms are a free-swimming marine single-cell organism. So they exist as a free-swimming state in the oceans. And then they have a metamorphosis stage in their lifestyle where they settle as a sessile tube worm, and they typically form on the hulls of ships. So big military vessels have some problems with these because they completely coat the underside of the ship, and they create a huge amount of drag as the ship. So they're everywhere, right? And ships in particular spread these guys to. The crazy thing is that this marine tube worm, when it transitions from this free-swimming state to the sessile state, it requires a bacterium, and it requires a specific bacterium. It's the Pseudomonas letuviolacea. I wrote that down because I knew I wouldn't. And so it requires this direct interaction between this bacterium with this free-swimming state, and then that interaction causes the tube worm to settle into, or metamorphose and change into, its sessile form, and it stays that as its sessile form for its life. The crazy part is that what Nick uncovered was that this bacterium had actually stole a phage tail fiber structure. And so you might have heard of these structures being type 6 secretion systems. Have you heard of these?
No, I haven't.
So type 6 secretion system is a bacterial secretion system, but it's basically a phage tail. So that end of of the classic Spacelander phage, bacteria have stole that entire gene and evolved it over the millennia, and they express it on its surface. They'll produce these, and then they'll express them on the surface, and they'll use them to deliver proteins and DNA into other cells. There's lots of examples of bacteria using them in competition, almost like a tit for tat. Like, you stab me with one of these, I'm going to stab you with mine and inject a toxin. But what this Pseudomonas one does, which is crazy, is it's stolen a phage tail, and it uses it to inject a specific protein into these free-swimming tube worms, and that protein is what causes the tube worm to enter into its sessile worm state, which it stays for its life, which is just absolutely crazy, right? You've got an entire marine organism that can't reproduce until it gets into a sessile state, and it requires a bacterium to inject a protein into it via a virus structure, and its whole life cycle has evolved and been built. It's just absolutely incredible.
Going in the direction of evolution, do you think which came first, viruses or bacteria, or eukaryotic cells, or prokaryotic cells? I'm sorry.
So I think Luca almost had to be a prokaryotic cell, some form of that. So I think cell came first. I'd see how to go a bit further and how and why. I think an amazing broad.
I know Forrest. Yeah.
Forrest. Yeah. So this idea I heard him give at a time, at least the basis. He thinks that viruses evolved from bacterial sex, and that was really cool. I really thought, and so I really like that idea. Sort of built on that. So I think a cell had to come first, but I think that cell, if you think about the Earth at that time, it was a big microbial soup, and life was probably very fragile. There was quite arguably really rare events that had to occur for life to key one. And they were probably really rare events. And for life to really spread and diversify, I think cellular life hits this barrier where it simply can't persist across space and time long enough to mix and share genes. And so I think for life to have evolved beyond that early microbial swoop, I think you need viruses. And from that, and what I mean by viruses at that stage, it's not a purely pathogenic element, but more a way that you can package up useful genetic information and store it, right? It's almost like a time capsule. You've got these stable protein packages that have packaged useful genetic information, and they're almost sent out in the environment like time capsules. They're going to persist. So if that cellular life dies off for whatever reason or gets outcompeted, the useful genetic information that it's evolved is spread across the environment until a point that early virus encounters another cellular life and it provides it with that useful genetic information. So I think that those early viruses were absolutely key in driving and allowing life to escape that really risky early stages.
Do you see any analogs? Like I thought about extracellular vesicles, and they seem virus-like. They package genetic material and other stuff. They can readily enter into other cells. They can regulate their genes. They can, who knows what else. And then I thought about, even conception, sperm infecting egg, and there's a whole person's created from that. Do you think that there's any relevance to seeing that or thinking that?
Yeah, it's a good point. I hadn't thought about the sperm and egg. It's a really interesting take on it. And you're right. I think there's lots of analog to that viral transmission, that nation. And, sex and mating, essentially, if you really want to break it down, the same conceptual process to a new organism, life form. And I think that's what early virusitic, I think, to transfer genetic, spread life into gene. And, to come back to somewhere along the way, that when the virus says, hey, life's already good now, I can be much more efficient by, I think parasitic virus came after the start as this sort of microbial sex. And at some point.
Do you think viruses are headed in some direction? It doesn't seem like they're becoming more virulent over time. It seems like they're going towards being commensal or even mutualistic or endogenizing into their hosts. But do you see that?
So, not really. I don't see them as going in a specific direction. I think one of the biggest misses with viruses is that they are pathogenic. That's true for a very, very, very small number of the viruses, right? It's just that right? Hey, we're living in, we'll arguably see. But the viruses, vast majority are symbiotic. They exist all across the whole spectrum of bioses. And so I don't think that. So no, I don't think that they're evolving in a specific way. They're becoming beneficial. If I was to distill my view of viruses, I think they cover sequence space. So if you look at, if you were to distill life down into its DNA, you could then think about different niches or environments that life could emerge and persist and be optimized within. And that's really, that's natural selection and selecting beneficial genes and combinations of genes to persist in environments in a cellular life where you package that. But I think viruses exist and cover on such a expanse of genetic information that they're almost searching and creating all of this sequence space that then cellular biology can co-opt and use to emerge and adapt and diversify. I don't know if that was
when you say they're searching the I guess this information space. Do you believe there was an information space to search? Most people say, "Oh, it's random and random mutations, and it's outside of all organisms. There just happens to be a beneficial mutation or not." But do you think that there actually is an information space that living things attached to, and therefore that's where their abilities come from? Like, if a cow is born, how does it stand within a minute of being born? No one taught it, or a baby suckles. Maybe all living things do attach to an information space.
Yeah. So I think there is an information space. It's not as here. You have to go. But I do believe there is, and I think cellular life has no way searching, and we're just too slow. You look at the adapt, and I think that holds true for a lot of a way than search and life can genetic at a scale that to explore. And so to come back and give this reason, viruses and masters can put and they will recombine, they'll chop and change, share genes, and probably 99 that's going to be a non-functional genetic space. It searched that space, it didn't work. But viruses have the number to be able to do that. It doesn't affect their replicative chance and success. And eventually they're going to stumble on this novel combination. We're going to come up with a new genetic space that we haven't been able to search before, and that may be able to be used and co-opted by cellular life to then push and expand that sort of genetic landscape.
I mean, in general, like how would you're expressing it already, but how do you see viruses playing a role in evolution and adaptation and maybe even speciation?
Yeah. I think that they are they're one of the major driving aspects of evolution. Of course, they arguably play a bigger role, but I think the emergence and transfer of this information is really driven by viruses. There's lots of examples of, if we take the predator example, right? So viruses predating on a specific bacterial population. One of the ways that the bacteria responds to that is to diversify, to create two species. And so now the virus can only infect one of those species and the other one escapes that viral pressure. And then that one, grows up. And so that's one way that viruses can drive diversification, because if any one form of life becomes too abundant, then that's exactly what a lytic virus is looking for in terms of replication. So viruses do drive speciation. I think one of the most fascinating aspects is recombination and how viruses recombine and how they shuffle and transfer genes. And I might give an example of this because I realize I'm getting a bit too hand wavy.
Yeah, I haven't asked. Yeah, what is recombination? How does it work? What's an example?
Yeah. Okay. So recombination is really, I would say simply it's the shuffling of genes, the recombining of genetic combination. And so viruses do this all. When you have a specific example, when you have multiple viruses in a cell, that gives the chance for those viral genomes to recombine and create an entirely new virus. And the example I'd give for this is in the human. And so in the human gut, specifically in the phagome, collecting and sequence the viruses of bacteria. Currently, if you can go to any person in the world, you can extract their DNA viruses from their gut and sequence them. And when you come back and you look at the sequence, between 50 to 90% of that sequence is completely unknown. And what I mean by unknown is never before seen, right? Never before seen combinations of DNA. And we've been doing this for, let's say, close to 15 years now. Sequencing power has massively increased over the last, 2 to 5 years. And still, we can still do this, and we can still sequence people's viruses, and we still find that between 50 to 90% have never before been seen. And so there's two arguments there, right? So one of the arguments is, well, we just haven't sequenced enough of the people. And so we just haven't really hit that plateau and that curve. And that could be true. And that might hold that, after another 5 or 10 years of really deep sequencing, that we might uncover enough of this information to start building that database. Or it could be that viruses are persisting at or adapting and recombining at such a rate that they're essentially creating new and novel combinations of DNA in every single person, in every single gut environment, and that they're really rapidly searching this sequence space, recombining, mixing, random mutations, all of those mechanisms that they're just constantly creating novel combinations of DNA, and we may never completely catch up and be able to search and sequence that. I don't know which one's going to hold true. I like the infinite combination, but I think it's it's still a bit in fantasy, and I think at some point we will fill in that sequence database. But it just goes to show the speed at which these can recombine and mix and create new genetic information.
Yeah, it's like they're a vast, omnipresent library of ability, and if you could harness it, I couldn't imagine what you could do. It'd be amazing.
Exactly. Yep.
Okay. Just a couple more questions. I mean, you've been rapid fire answering a lot of.
No, it's fun. It's really good.
This one's always concerned me. I don't know. Throughout all time, I'm just going to throw a number out there. I don't know. There's been 10 to the 50th successful viral infections of something. 10 to the 30. I don't know. We'll just throw out some huge number.
Yeah.
But if viruses, a lot of them are non-motile and they're just passive supposedly and floating along,
Yep.
and they're like 50 to 100 nanometers, and they're encountering this gigantic host, a person. So
Yeah.
there's this tiny, tiny thing in a huge field of host. How do they find their targets? Quadrillions and, 10 to the 30th times over time. How does this happen so often if there's no guidance system, if there's no sensing, if it's just, they're just floating along?
Such a cool thought experiment. I love the scale. When you try to think how to encounter, you can't even comprehend. Such a cool, really love. So what I'd say is that as big as that divide is between the size of a virus, the numbers of viruses is so many orders of magnitude more than that scale difference in size. And so it's estimated there are 10 to the power of 31 phages on the planet. They're the most abundant biological entity on the planet. And no one can even begin to comprehend 10 to the power of 31, right? We can barely begin to comprehend a billion, right? You look at some of these graphics that people show, the wealth of Jeff Bezos compared to, Bill Gates, and the scale is astronomical. And that's one order of magnitude, 2 orders of magnitude. You're speaking about 10 to the power of 31, right? So that scale is beyond our comprehension, right? And that's one thing that I'd really say. We cannot even begin to comprehend the numbers game that viruses play. And this comes back to that whole genetic landscape, but I won't go back to that. So to answer your question, how can a virus that's so small ever overcome and infect and find its host? If we go back to a phage, right? A phage floating through the gut, which is a hugely complicated and diverse ecosystem. How can a single virus find a bacterium? And I'd say that it can't. A single virus could never, right? It's the chances of doing that would be infinitesimally small, that it would probably never happen. And so the only way that it can happen is by having these ridiculously large numbers, huge numbers that we can't. So maybe I'll come at it from the other angle. How many viral infections are successful, right? And I don't think we'll ever know the answer to that, but it will be infinitesimally small, right? If there's 10 to the power of 31 viruses on the planet, there's probably, at least 90% of those are failed infections or viruses that are just sitting in the environment somewhere, hoping and waiting to be used. And so viruses, they overcome this problem of scale across space and time with numbers that we can't comprehend. And when you flood a system with that many numbers, encounters that were borderline impossible become borderline, they're almost absolutely going to happen. And so that's how viruses are, and that natural sort of diffusion and persisting in that environment is going to drive these chance encounters, and that's going to drive more and more of these viruses to persist. Can I give you one more example of that? And this is from some of my research I did in my postdoc with Forrest, and it's a cool example that I wanted to touch on. And it comes up, it talks about this, like how does a virus find and encounter a host? And what we looked at was in the mucosal surface. So in the gut, you've got your cells, and those cells produce a mucus layer. And that mucus layer is really critical to support your microbiome. And what we found was that specific phages would bind and stick to that mucus. So they're viruses existing, diffusing, just randomly. And then they'll encounter a mucus. They'll encounter a mucus strand, and they've got specific proteins on their head, which allow them to stick to that mucus. It's a really weak stick. So it'll stick for a little bit, and it'll detach, and it'll keep diffusing. And it's sort of this on-off motion. And what we found initially in our first paper was that by sticking to that mucus layer, they protected those mucus cells from bacterial infection, right? So you could think of this as like going from a 3D space into a 2D surface where phages stick to that mucus. And if a bacteria tries to come through, they're now entering a higher concentration of phages, and that they're more likely to be infected, which is a really simple solution to that. But then we got really detailed into the math, and we started looking at how these phages diffused. And so actually watched them in real time on a microscope, and we tracked them in water, and we found that they diffused normally. And then we tracked them in mucus, and we found that they diffused subdiffusively, which is this really complicated mathematics behind diffusion. And to make it really simple, what we found, or what we showed, was that phages would diffuse normally, and then they would stick to a mucus, and then for an undefined period of time, and then they would diffuse again. And it was this sort of on-off motion. And we started looking at like search strategies. So have you heard of a Levi flight?
No, what is it?
No, so a Levi flight is a search strategy that certain animals will use. So they've shown this in an albatross, where an albatross will fly in this specific manner to search for prey in it. And it's a mathematically optimized search strategy to find something. They've shown this in honeybees, in insects, even in hunters and gatherers from Africa use this search strategy known as a Levi flight. And so we hypothesized that by the phages sticking, they exhibited this subdiffusive motion, and that this was a strategy. It was a mathematical search strategy that allowed the phages an increased chance to infect a bacteria. So I realized that was really long and winding. So just to recap, so we found that phages stuck to mucus, and that changed their motion, their diffusive motion, and it gave them an increased chance to encounter and find a bacteria. And we made an analogy that was similar to a search strategy. So even though
boiling this down, it wasn't random motion. It was a heuristic that is echoed by other creatures in nature.
Yep.
And even just mechanically,
yep,
it was like you're in the ocean, you're grabbing onto flotsam and moving, using anchor to anchor.
Yep, exactly. Yep, and it was
amazing.
Yeah, it was super cool. It was a huge amount of work. Mathematics was really a project.
That's so cool.
Yeah.
If you would, I just have 2 or 3 more questions, and then
cool.
For a given disease, if I'm the first person to get it, if I'm the first person to get a viral infection, let's say, I don't know, from a monkey, and I just label myself number 1,
yep.
And then I infect, my brother, he's number 2, and then he infects his girlfriend at number 3, and let's say someone's number 20 in this chain, this passaging chain. I don't even know what you call it. What do you think? What do you think the virus would look like in them versus number 1 or number 10?
Yeah. So it would have used the term micro. So it would have optimized itself to replicate in the humans, right? Within patient 20. And that's on a genetic level, that's probably going to be a bunch of point mutations in the virus's genome. That's going to cause. These aren't massive jumps, right? These are micro jumps. They're really small changes that the virus can make to maximize its replicative efficiency. And to go back to phages, sorry to keep doing this, but a phage is a virus, and we actually do this with phages all the time. So another aspect of our lab that we're working on is phage therapy. So the use of a phage to combat a bacterial pathogen, specifically ones that aren't responding to antibiotics. And we go out in the environment all the time and we find novel phages that can kill these superbugs. And what we find is that when we first isolate that phage that can infect a superbug, is that it's not that great at killing it, right? It might, not produce that many viruses. It might be quite slow. And so what we do is we passage that phage on that host multiple times. So we'll let it grow, overnight for successive days. And what we'll find is that at the end of that virus is better. It's much more efficient in infecting that host. And what's happened is you've gone through these like microevolution steps, these point mutations that slightly optimize the virus to that host. So I think these are micro jumps that allows the virus to almost, speciate to better infect its host.
Makes sense.
Yeah.
Has anyone done this with bacteria and looked at the population of virus, population of phage, and sequenced all of them to see the differences, and then you passage them, sequence them all, and see how the populations change and grow and
all the time. So it's this field of experimental evolution is really cool. There's been lots of classical examples of a single phage and a single bacterium and looking at how they co-evolve with each other. We're actually doing an experimental evolution experiment right now. We're wrapping up on that phage and mucus. Rather than evolving the phage to the host, what we've said, can the phage to better stick mammalian mutant it can accumulate mutations that increase its fitness. So it's actually, I would say that the use of in the field of it has been critical, probably their generation side, that they can really rapidly explore this state and sequence and figure that out.
I wonder if we understood it well enough, you might be able to use viruses as like a biological AI to search the information space for an answer faster and better than we could with just computers.
That's a super cool thought. I'd never thought. That's pretty awesome.
Well, you gotta figure it out and make something like that. That would be really cool.
Yeah, that's really cool.
Oh, well, good. Well, Jeremy, this has been awesome. It's like a tour de force, this interview. I love it. Thank you so much for doing all this.
No, that was really good, Richard.
Is there anything else I'm going to ask you? Or if not, I'm going to ask listeners where they can find out more about your work, because there's so much there. It's amazing.
Yeah, no, I think we covered a lot. There's nothing top of my head.
Okay, and where can listeners find out more about you and your work?
Yeah, so Google Internet's probably easiest. You can Google my name and our lab will come up. So our lab is at Monash University in the school, and the address is barlab.org. I'm also on Twitter, so at Jeremy J Bar. But yeah, usually email. More than happy to.
Okay, Jeremy, thanks so much for coming and contributing to this book, and you got a lot of great examples. So I appreciate you being here.
Yeah, no, thanks, Richard.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.